Hello everyone. Welcome to my channel Biochemistry Insights. As you all know, we are doing video lecture series on the subject AMR. This is my third video. Video number 3 on AMR, and the topic is Classes of Antibiotics. As you all know, we will be dealing with this particular topic in much more details later on. Therefore, this is a short video which will give you an insight into the classes of different antibiotics available. We can classify antibiotics in two ways. Number one, on the basis of their mechanism of action and by its chemical nature. The first category of antibiotics, they are classified or they are grouped according to how they act on this organism. Sometimes they act on the cell wall, sometimes they act at the level of cell membrane, and even they can act at or inhibit certain fundamental function inside the cell. So let us see the group of antibiotics which come under this category. First group of antibiotics is the cell wall synthesis inhibitors. They prevent bacterial cell wall formation leading to cell death. Examples are penicillin, cephalosporin, vancomycin, bacitracin and cycloserine. These antibiotics, they will block either the enzyme involved in cell wall synthesis or they modify the target of the enzyme which is involved in the cell wall synthesis. So, some way or the other, they will block the cell wall synthesis. And as you all know, cell wall is very important. If it is not synthesized properly, obviously the cell would die. The second group is disruptors of cell membrane integrity. Just like cell wall, cell membrane is of utmost importance. Any substance which can disintegrate the cell membrane can actually cause damage to the cell. This can lead to leakage of essential components inside from the cell, and examples are polymyxin, nystatin, amphotericin B. Now there can be antibiotics that can inhibit very important processes inside the bacterial cell. For example, inhibitors of protein synthesis. Now, protein synthesis inhibitors, some of them whose action can be reversed, and there are some whose actions cannot be reversed. They permanently damage the cell. So first let's see if protein synthesis blockers which can cause reversible action. So they block bacterial ribosome. They stop protein production without killing the cell immediately. And the examples of this type of antibiotics are tetracycline, erythromycin, chloramphenicol and clindamycin. Now we also have protein synthesis inhibitors which are irreversible in their action. That means once they have done the damage is forever. So they bind the ribosome, cause faulty protein production leading to bacterial death. Examples, aminoglycosides such as streptomycin, gentamicin. Next is inhibitors of nucleic acid synthesis. So these group of antibiotics, they disrupt DNA or RNA synthesis. So once DNA or RNA synthesis has been blocked, the bacterial growth will be stopped, and example include rifamycin or rifampin, quinolones, etc. So here we see that antibiotics can be classified on the basis of how they act on the organism and which particular component of the cell or which particular process of the cell gets inhibited. Now, we just saw that the first class is based on action of the antibiotic on the cellular components of the cell itself. The second class is actually based on chemical structure of antibiotics. So here also we have groupings depending upon the basic chemical structure of the antibiotic. So remember, the earlier class was grouped according to the action, and the second class is based upon the structural aspects of the antibiotic. Here, the first important group is called beta-lactam antibiotic. Like penicillin, these antibiotics have a core structure that include a thiazolidine ring and a beta-lactam ring. So here you can see that this is the structure of penicillin, where you can see the basic structure, okay, which are crucial for its antibacterial action. This structure is shared by all penicillins and is known as 6-amino-penicillinic acid or 6-APA. By modifying the side chain attached to this core structure, different semi-synthetic penicillins are created. The second group of antibiotics is aminoglycoside antibiotic. The aminoglycoside antibiotics, they contain one or more amino sugars linked to an aminocytitol ring by glycosidic bonds. These are broad spectrum antibiotics. In general, they have greater activity against gram-negative than gram-positive bacteria. Examples are the common antibiotic streptomycin, which has been isolated from a strain of Streptomyces gracias. The third group is tetracycline antibiotics. Tetracyclines have a ring system of 4 linear 6-membered rings. So here you can see these are the 4 rings, and they are characterized by a common octahydronaphthalene skeleton. They are potent, broad-spectrum antibacterial agents, effective in combating the bacteria, mostly the gram-positive and gram-negative aerobic and anaerobic bacteria. Example, doxycycline and minocyclines. The fourth group include polypeptide antibiotics. The compounds that have complex polypeptide structure, these are resistant to animal and plant proteases. These contain lipid moieties besides amino acids that are not found in peptides of animal and plant origin. Example, bacitracin, polymycin, amphomycin, tyrosine, and vancomycin. So here, up till now, we have seen that we can have organic molecular structure. We can also have carbohydrates-based antibiotic, and these polypeptide antibiotics, they are usually lipid and protein components they have in their structure. Apart from the ones which we have already discussed, there is a fifth group which is known as macrolide antibiotics. These are a class of mostly natural compounds with a large macrocyclic lactone ring to which one or more deoxy sugars are attached. The lactone rings are usually 14, 15 or 16 membered. Macrolides belong to the polyketide class of natural products. The macrolides are commonly administered for respiratory, skin, tissue, and genitourinary infections. Examples, the very popular azithromycin, clarithromycin, and erythromycin. The last class is lincomycin. The lincomycin antibiotic is a lincosamide antibiotic obtained from actinomycetes, streptomyces, and lincolensis. The ability of lincomycin to penetrate into bones adds to its qualities. So this is very important. Lincomycins, they can penetrate into bones. So if you have some infection in the bones, so these are the class of antibiotics which we land up to. It gets promoted in the chemotherapy of bone and joint infections. Example, lincocin and lincorex. This is the structure. representation of lincomycin antibiotic. So we come to the end of this video. Thank you for watching. Learn more about AMR and watch more of these videos. If you liked and enjoyed this video, please don't forget to like, share and subscribe. Thank you.